¿Me puede escuchar ahora?
Sí, sí, ya.
Ah, perfecto. Muy buenos días. ¿Cómo se encuentra?
Buen día, muy bien, gracias.
Perfecto. Ya vamos a entrar en unos 15 minutitos,
Sí,
pero qué bueno que se conectó temprano, así ya pudimos probar el audio. No sé si usted pueda ver mi pantalla.
sí, veo como, que será como un proyector, o sea, es una proyección.
Sí, sí, es el auditorio.
Ok, sí.
Así es como se va a ver más o menos. Y déjeme ver nada más. Em, perfecto. ¿Podría probar a ver si puede compartir pantalla para que se vea?
Sí, permítame. Ahí
Ah,
ya,
muy bien.
¿sí?
Sí, perfecto, perfecto. Así está muy bien. Muchas gracias.
Sí, no, al contrario. Yo creo que, no sé si a lo mejor al inicio de la plática pueda yo prender el video, pero durante la ponencia preferiría mantenerlo apagado por cuestiones de mi conexión de internet.
Ah, perfecto. Sí, sí, sí,
¿Sí?
claro, 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 se entiende, sí. Sí, muy bien. Sí, ahorita pues son todos los problemillas de
Sí,
comunicarse a distancia, ¿verdad? Claro.
sí, sí, no, no quisiera que fallara. Generalmente cuando tengo video y presentación he tenido algunos detalles, pero bueno, fuera de eso no creo que exista mayor problema.
Perfecto, muchísimas gracias. A ver,
Sí,
yo
al contrario.
ahorita, yo estoy aquí, bueno, me voy a ver un poquito de lejitos nada más,
Ah,
para que
sí,
me
sí.
vaya, para que me vaya conociendo aunque sea. Muchas gracias.
Sí, no, al contrario, gracias a ustedes.
Sí, a mí me pasó su contacto la doctora Esperanza Torres. Este, ella es mi esposa y está ahí en
Ah,
geología también.
de verdad,
Sí,
qué pequeño es
ella
el
está,
mundo.
sí, claro, claro. Entonces ella de hecho, cuando estábamos buscando así gente para el evento, pues a mí me dijo Esperanza, yo conozco a una investigadora que está haciendo biotecnología ambiental. Y dije, pues queda perfecto para la semana.
Qué bien. Sí,
Sí,
se me hizo curioso, digamos, pero digo, aquí el reto era es que hay que hacerla en inglés, pero fuera de eso, muy bien.
sí, 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 claro. Ahorita porque estamos nada más nosotros dos, pero ya cuando empiece el
Sí,
evento, pues sí, nos echaremos a hablar en inglés, pues.
a ver qué tal, ¿no? Ah, no es que.
No, claro, todo va a estar bien, todo va a estar bien. Va a haber cosa nada más de acostumbrarse tantito y ya.
Sí, sí, sí.
El, el, también la liga, mire, voy a aprovechar también porque se va a transmitir también por YouTube Live,
Ok,
por el canal aquí de la Universidad de Arkansas.
sí.
Entonces, déjeme ver, este, porque me dejaron aquí las instrucciones de cómo hacerlo. A ver, en vivo en YouTube. ¿Cómo que no? A ver. A ver, un momentito, porque por supuesto tenía que haber problemas, ¿no? Con esto. Yes, yes, you come in. Hello, please sign the attendance sheet. Okay. Okay. Ahorita vengo, doctora Aurora, nada más tengo que resolver esto del YouTube. Me voy a sentar unos 5 minutitos, ¿eh?
Sí, sí, no se preocupe, adelante.
Utilizo el tuyo, ¿eh? Un segundo.
Sí, claro.
¿Tienes el Google Chrome?
Google Chrome.
¿O algo?
No, mira, aquí me voy a más
¿Sí?
y me voy a YouTube, ¿no?
Sí.
Y aquí me manda.
Use another account.
Sí.
Ok. Academic. Sí. Let's see. .mx. Ay, ¿tienes la password?
Sí.
Es QRO.
Guión, sí, cierto.
Guión.
2022.mx con mayúscula.
X
01. Sí, se me olvida esto, perdón, disculpa.
No. Imagínate, no pasa nada.
No, ¿verdad?
Ya estás, pones el nombre
Sí.
y go live.
Muchísimas gracias.
De nada.
Hey guys, welcome. Please, if you haven't signed the attendance sheet, do it. Please. Okay, guys, welcome. This is our second talk for the same week. Please, if you haven't signed the attendance sheet, do it. Today, we're going to have a very nice talk by Dr. Aurora Espadas. So, Dr. Aurora Espadas is a biochemical engineer, and she holds a PhD on environmental science and a master's degree in environmental engineering. She's currently working as a researcher at the Geology Institute of Oman, focusing on bioremediation, water treatment, and low-cost materials for hybrid processes and constructed wetlands as nature-based solutions. She also teaches some courses and selected topics for undergraduate and graduate students, and she's also a member of the National System of Researchers Level 1. She has published several research articles in peer-reviewed journals, and she collaborates in research projects with countries such as Australia, Chile, and the USA. She's also the chair of Young Water Professionals Mexico, that is part of the International Water Association, a platform to share knowledge and promote participation of young professionals and students in research and development of solutions regarding the water sector. So, Dr. Espadas is what we can consider environmental biotechnologies. So it's a very interesting topic for you, biotechnology undergrads to understand, to get close to, because not everything in biotechnology has to be research regarding the pharmaceutical industry. You know, there are some environmental approaches that can be very interesting for you. So please help me give a round of applause to Dr. Aurora Pat. There she is. So the audience is yours, Dr. Pat Espadas.
Yeah, thank you. Thank you all. And first of all, I want to thank you for the invitation to take part of this STEAM week. For me, it's a pleasure to be here, and I hope that you can enjoy this talk, which is called Biotechnology, and I will give some options for the treatment of wastes that contain potentially toxic elements. So let's start, and I will start explaining the term which is potentially toxic elements, which is PTEs, and this term is adopted as an alternative to the title heavy metals or toxic metals or trace metals, and it's also an environmental context to use it. And also it can encompass both essential and toxic elements in heavy metals. And here we need to remember that all elements are toxic, but beyond a certain threshold value. You know, it still toxicity varies with the nature and also with the chemical species of some element, and it depends on conditions such as nature and other environmental factors, as we can see here. For example, while an element can be essential for plants' growth, for example, beyond a threshold, it would become toxic for them at all, okay? So let's see an example. An example of this, if we think about lead, we can think about lead as a toxic, but it depends on the chemical species. And here in this picture, what you can see is lead as a free cation, such as lead 2, but also as a galena mineral, which is solid, which it has crystal structure, which is also containing sulfide lead, and it is not a lead alone. So they are both contain lead, both of those species, chemical species, but they are quite different, and hence the environmental behavior and the mobility will be far different from all of them. So it can occur with not just with lead, such as this example that I'm showing you now, but it can happen with other cations and with other elements as well. So we need to look at the chemical species. And also, let's think about the PTEs, the potentially toxic elements in a certain environment. And we are going to talk about presence and concentration. And as I said before, chemical species is important, but also the concentration. And we can think now that the mere presence of an element does not confer it to be used with connotation of pollution or toxicity. So we need to look far beyond that. We need to look at the ionic speciation, as I said before, and which is a key aspect controlling mobility. We can look at the recalcitrance, the persistence, and also which is the mobility of these elements, which turn it on highly toxic in one form, while they may be essential in another. They're different here. For example, the figures that you can see here is from iron. At the bottom of this picture. So iron is an essential nutrient, for example, for us, because we need it for some metabolic processes, and it's essential, but beyond a threshold, it can be toxic, well, with an excess, and depends on the level in the body, which can be tightly controlled. So an environmental example can be the eutrophication, which is the picture you can see on in the environment, which is characterized by excessive plant and algae growth, which is in green on the picture. Is an this phenomenon increases the availability of one or more limiting growth factors needed for photosynthesis, such as sunlight or carbon dioxide or nutrient fertilizer. So it can occur naturally, but human activities have accelerated the rate and extent of this phenomenon called eutrophication. And it also has consequences which can be dramatic for drinking water resources, for fisheries, and also for recreational water bodies, such as you can see in this photograph. So as we have already mentioned, the mobility and definition of this term in this context, an environmental context, it is about the dissolved methyl forms, which are actually the responsible for environmental risk. And it can also include some aspects such as parameters like pH, the oxidation reduction potential, the presence or not of organic matter, and the cation exchange capacity. But it can also depend on nature or environment, such as the origin or the source of the contaminant. And it can also be influenced by conditions like temperature, humidity, the presence or absence of oxygen, and also the presence or absence of microorganisms. As you can see here, there is a lot of parameters, a lot of variables that are taking part on the response of the metals or in the elements and the changes in the environment. So it's not as simple as we can imagine. It is a more complex process once the metal is released to the environment. So it is changing according to those conditions. And we need to determine which are the species and which are the mobility and which are the environmental effect of these elements. So let's see some pictures to illustrate those problems. So talking about environmental aspects, there is some examples of pollution. And in this slide, what you can see here is the difference between recyclable and biodegradable. We can see here, for example, a can and also a banana peel. So some wastes can be completely biodegraded, such as the banana peels that we can see in this picture, while other wastes, such as those containing metals, persist over time. So it's a far different one from another. And we can look for different technologies or options to be treated. So this type of contaminants may be produced due to various nature phenomena, but it also are related with anthropogenic activities. So there is mainly industrial activities who have led to a large scale contamination of the environment with toxic metals. So they are well related with anthropogenic activities. And regarding this kind, but also biodegradable or not biodegradable materials, let's see these examples. So the difference we can see, we are going to see here in these problems which derive from contaminants. So in this slide, what you can see here is an example of the sargassum seaweed, which is affecting Mexico's beach. So the Caribbean coast of Mexico is really affected by this natural, this nature here is a natural phenomenon which we can see here, but it is also not a good thing to do. Also, if you go to the Caribbean coast and you can see this picture, it is not a good view to see. There are even some problems, but it is nature here. This, we can just make some technologies, we can clean the beach, and we can just try to avoid the bad things that can occur about this phenomenon, but I mentioned that it is nature here. So let's see another picture here. And what we can see here is another beach, which is also affected by some problems. But in this, it is natural occurring. What do you think? It is plastic waste. It is also in Mexico, in another beach, but there is not related with nature. Yes, it is. It could be avoided, but it is also some beaches a very natural host. So it is not nature here, and it cannot be treated like the other, like the seaweed. With seaweed is nature. Here it is for anthropogenic activities at the beach. So let's see another one. Here, for example, there is, so here we can see volcanoes emissions. So there is some nature phenomenon, as I said, such as volcanoes emissions, like during these periods of intense unrest or eruption, volcanic emission rates of metals such as cadmium, copper, zinc. It can be equal, or in some cases, it can be also orders of magnitude greater than the anthropogenic emissions from entire regions. But volcanoes, for example, is one of the largest natural sources of many metals, but it is also natural. You know, what we can do here is just to let it happen, okay? But it is also a natural source of many metals, also are one of the largest ones. But let's see in this picture, another source of metal. So here, this is not natural, it's anthropogenic, and it is what we can see here is the acid mine drainage, which is due to waste from mining sites. Is the formation of highly acidic water, which is also rich in heavy metals and also rich in sulfide, but it is also not good for the environment. It can also be related with health problems, and it, of course, it is not desirable to occur, but we can see those scenarios on environmental concern in some places. So it is not great to occur. So we can see here nature versus anthropogenic things that occur. In both of them, we have metal release, but there is some difference about the source of these metals release on the environment. So, and what happens once it's released in the environment, it is linked to metals mobility, as I said on some slides before. And mobility, it is the main cause of problems because there are different routes of transportation within the environment. There are also, of course, factors that help to find solutions to contamination. And there is also complex pathways that they can follow during the travel, we can say, on the environment. So anthropogenic as well as natural sources of pollution, all of them are just releasing metals. And when it is present on different metrics, environmental, such as water or soil or atmosphere, they just can go through those phases and experiment different transformations. But we just can remember that that metals, for instance, they cannot be degraded. They can also be transformed or converted into some chemical species or another. So it's important in which species the metal it is. So it is not the same as we said before. A free cation, it is different from another sulfide mineral or another chemical species, because it affects in some different or in different ways to nature, to plants, to water, or the organisms that live in there. So, and now what is the microorganism participation in this? So the presence of microbes, it's related or it's known as a key or crucial factor, since microbes have evolved to deal with toxic metals using several mechanisms that we can see here summarized. And also because microbes can transform or biotransform the chemical species of metals. So microbes can change the oxidation states of metals and hence promoted the formation of soluble or insoluble compounds. Such as this, we can mobilize or immobilize the metals depending on the goal and also depending on the strategy to follow. For example, here, I think that it's this figure it sounds a little bit complex, but what I want to show you here is that there are different things that happen or that are promoted by microorganisms. So we can see a microorganism or maybe a bacteria like a black box, you know, and also we can see it with a naked eye, but we can see that it happened a lot of things on these microorganisms. So a lot of reactions that can be metabolic and all of them that can be promoted to lead the transformation of metals. So some of them that you can see here is bioleaching, bioaccumulation, biosorption, bioprecipitation, and all of them, or bioreduction, all of those reactions that are promoted by microorganisms affect their chemical species that we can talking about before, and also the pathway or these routes during the using microorganisms can be chosen, and also can be we can take advantage of those reactions promoted by microorganisms. So not all of the microorganisms can do everything what we are seeing here, but one of them can be, for example, one type of microorganisms can make bioreduction. Another one can bioaccumulate some metals. Other ones can bioprecipitate or biosorb. So there are different microorganisms that we can use for one purpose and just follow that route to deal with the contamination, or just to try to change the metal mobility in the environment. So this is the importance of use microorganisms in this context. And this is the main thing which my talk will be centered of. So. But before we start talking about this one, just let's see some reasons of what we have problems with metals now in society. So just before to end this, I have to see that bacteria can catalyze different reactions and it affects metals, and that's why we use with biotechnological purposes. So the development of remediation strategies is centered on this, on one of those reactions, so that we can see here. So, but the scenarios that we can look now in our environment, let us think on consumption or modern era. So the metal use has increased through the years because of technology development, mainly because of technology development is one reason for this. So in this graph, what we can see here is the use intensity in products through the time. For example, before many years ago, there was not such technology that we have now. So now we can use a lot of things, including clean energy technologies, like solar panels and more. So and those technologies development and those social commodities that we can use need metals to be achieved. So it is a reason for which the consumption, the metal extraction has been increased, and it is also thought that it would increase more in the next years to come, but it can also be decreasing. It depends on which the society goes, and also how to recycle the resources that we already have. But the number of metals we use and those technology development can also lead to the increase of metals in our daily life. So that's one of the reasons that we have here. And also a metal to be used, for example, that we here in this graph, has to be mined, it has to be extracted, and it has to be separated from the natural environment, yes, to be used for a product, for example, for a cell phone or for a technology device. You know, there is a lot of operations that are associated with metal extraction and the processing. And also there is a waste products that are just increasing, as for example, as a cell phone model is just passed from the old one to the new one model. We want the new one, you just throw out to the waste the last one, and we need more the new model, the new technology. And it is also common waste products that lead to these wastes to be increased, and also the processing of those products. So we don't need it also anymore, the old ones. We need new ones. So I mean, it is like a cycle of wastes here: wastes to extract it, wastes to produce, wastes to discard those products. So as far as I know here, we have some sources to the wastes. One of them, we can also produce wastes or residues, of course. There is also those that comes from industry, more exactly for mining, or to produce the commodities or the products. We can also hear such as industry, agricultural or atmospheric depositions is some sources from which potentially toxic elements can be found. So there are a lot of things that we can do here. But let's go back, such as throwback Thursday, which in this case is not a happy memory here, but it is also the history of Mexico, meaning it's an activity that was extended like or date from 500 years ago, from the pre-Hispanic period, I think, when mines were established for gold and extraction during the colonial era. So those mining activities in the past, it leads to a vast mine waste legacy. This waste is present ongoing challenges regarding their management. So, however, until now, it is unknown how many sites contain mining waste and tailing deposits we have now. This map that we can see here is just recent efforts that we have made to map all of those sites. And as you can see here, the deposits that it is expected to find, those passive and also old wastes, I mean, past, not active mines. It just deposits that are from mining activities from the past, I mean here. So there are a lot of them, and also we are not sure that those are the unique or that those are the exact number that we have now here, but there is just an approximation of the wastes, of the abandoned mine wastes that we have already in Mexico, and it is not a good scenario to see, since all of them represent a source of environmental concerns, since potentially toxic element could be also be here. As you can see here, we can say like lead or gold, zinc, aluminum, and all of those metals that cannot be good for the environment, and it is also for health problems associated. So here, let's call this mine waste legacy, or these wastes received also Nahuatl word in Mexico, which is jales. This waste is known as jales, derived from Nahuatl shali. It means fine sands, and they have some characteristics such as low pH. They also contain potentially toxic elements. These lands now contain vegetation. They are susceptible to erosion. And as I said before, it is from the past, mining activities, because nowadays there are some regulations to govern exploration, extraction, and closure in mines. For those scenarios, it comes from past activities, and they are characterized by those factors. So we need to develop some remediation strategies to try to change these bad scenarios that we can see now here. So these photos also that I can show you here illustrate some areas that are located in Sonora State, which is where I live now. As you can see here, mine wastes, they are not also solids, they are also acid mine drainage, because they are also damaged water. So we can see solid, we can see water, and they're also part of the landscape. Here the landscape cannot look like this, but there are a lot of those scenarios, I mean, of these pictures that we can see just in this state at the northwest of Mexico here in Sonora. But one of the reasons that encourage the study and the development of possible remediation strategies, which are the points that I will try to address regarding the research efforts on this area, and some of the investigation that I carried out now in one of those scenarios, on 3 of them that I'm studying right now. So let's see some of this approach. So I will present, like I will be between 2 or 3 approaches of cases that we are using, to try to find remediation strategies for those sites in this area. So the first approach or thing that we can do is try to find microorganisms that would be able to grow or live in the site, which take back to the photo, and you can see, oh, that there's nothing living here. You know, it's sterile scenarios, but microorganisms, I said before, have lived with those environments. So we try to look for those that we can use to achieve metal removal or immobilization. So in this slide, you can see traditional ways to do it. So let's see just at the Petri dishes. So what we are doing here is to take a sample from the site, from the environmental site, and try to isolate the organisms. So in this case, in the Petri dishes, we are trying to isolate fungi on agar Petri dish. Here, the point is to use an adequate growth media and transfer, that many transfers until isolation is achieved. What we do here is to start from diluted solution of the solid samples, used as we do it with soil samples, but in this case there is the material, the waste that we start from them to isolate the organisms, and the experiments can be performed with the agar, both modified or spiked with different metal concentrations to observe the tolerance of the fungi, and also change after that to liquid media and start to develop a biotechnological process based on these microorganisms isolated from the site. So we have good expectations here because those organisms are able to tolerate a wide range of metal presence. So that's one way that we try to look to find those microorganisms. The other approach that we can see here is the isolation, but not fungi as in the first case, but of bacteria. And one of them that I also studying now is the sulfate reducers bacteria, which also are anaerobic microorganisms in anaerobic conditions. And it is also a little bit the same, but in this case, it's an growth media prepared according to the requirements of this type of microorganisms. And also the inoculum is also the sample collected in the field. We just put an amount of this inoculum, which is the sample collected in the field, and just try to incubate it under these conditions and try to find if there are microorganisms that can show some activity during the incubation. As there is not a short experiment, it's a long-term experiment, because sometimes to find microorganisms or microbial activity in those experiments, it can take too long. It is not like one day, it can take months or even a year to try to find out. It's also a good way to be patient, try to find the activity of microorganisms in those conditions. I couldn't find with my students sulfate reducers on those samples. We need to wait like more than a year to see a clue about that microbial activity. While fungi was a little bit faster, we found different types of fungi living on those samples that we collected, different of them. So we tried to, that we do was to work with 2 of them and try to find how they tolerate different metals and concentration, and that's what we are doing right now. Well,
that's one approach to try to find microorganisms from the real samples to use after that in some biotechnology approach. So let me talk a little bit more about the microbial sulfide reduction. That's the thing, or the microorganisms that we are looking at this sample. So microbial sulfide reduction, as you can see here, is a metabolic process that leads energy, of course, to the organisms. But here the main reason is that they use sulfide, and microorganisms reduce it to sulfide. It is sulfate to sulfide. So sulfide is released, and once the sulfide is released, this sulfide, which is a biogenic hydrogen sulfide, it can also derive another reaction that is mainly a physicochemical reaction, in which this sulfide can precipitate other metals, because they lead to sulfide metals, as we can see in the second reaction of this slide. So in the first one, we have the microbial activity, biological activity coming from the sulfide to sulfide, with, of course, in the presence of organic matter and microorganisms. And those microorganisms are obligate anaerobes, but they are widely distributed also in terrestrial environment or in the marine environments. But we can, as I said before, we can find it in those wastes. And the next reaction is the use of that biogenic hydrogen sulfide to produce a sulfide metal. So in this way, we can lead or transform the free metal to sulfide, which is more stable to the environment. It can be immobilized because those fractional sulfide metal has low solubility, and they also precipitate from the media. So it can occur with, for example, with metals such as zinc, copper, nickel, cobalt, iron, and lead, cadmium, and all of them. So this is the reaction that we promote to occur, but we need the microbial substrate microorganisms. So, and those are examples of those sulfide minerals that can be produced from potentially toxic elements. For example, from lead, which is the one I show you at the beginning of this presentation, the galena, which is now here is lead sulfide. We have this valerite, which is zinc sulfide, pyrrhotite, which is iron sulfide, pyrite also. We have the realgar, which is arsenic sulfide, and we can follow the list of those minerals that can be formed. So is a way to change the chemical species of the metal by promoting this reaction by sulfide reducers microorganisms. So as we can see here, we now have a solid which can be separated, and that is also more stable, environmentally speaking, for those potentially toxic elements. So just following the example that I can show you is that we collect the samples from a mine tailing. We make some microcosms activities of biological incubations during controlled conditions in the lab, and we also followed some parameters that was measured, such as, of course, sulfide, sulfate, and organic matter in form of lactate, and also the characterization of the product obtained by some analytical techniques to determine whether the potentially toxic element was mobilized or immobilized. So what we are looking here is not just the microbial activity, but also the change of the metal species on that we have present on those samples. So in the next slide, I will show you the results of those experiments. Of course, we make characterizations and analysis before and after the treatment of the same samples. And what we are looking here is different phases. I mean, there is a part of the potentially toxic element that can be in the water extractable phase, another that is more bounded to some carbonates or iron oxides or to organic matter, but the phase that we wanted to promote is the residual phase, which is fairly immobilized. So we are interested in change the water extractable presence to residual phase. And that's what we are looking here in the samples before the treatment. The water extractable phase, it was the more present on the samples, and after the treatment, the residual phase was instead the phase that was present the most. So we could change the speciation from ionic free presence to a residual phase promoted by sulfide minerals formed under the sulfide reducing treatment. So it was carried out by sequential extraction procedure performed to those samples. And the results were that we obtained here really encouraged the study. So we were able to change and to promote more the residual phase instead of the water extractable that could be mobilized better in the environment. So the treatment by sulfur reduction in those terms, it was successful for those samples. And it was one case that I can show you now here. So the second one is just theoretical, and other thing that we are using now here is one approach called nature-based solutions, which is it's not an old term, it's most like a new one, but it refers to action inspired by, supported by, or copied from nature, which are implemented also for to try to for remediation purposes, we mean, and they have also are characterized by being cost effective, provide social, environmental, and economic benefit, and it's like a new way to develop the strategies, because we are not just looking for environmental condition, but also we need to look at the social aspect, and we need to look also for economic benefits. So it's like a holistic approach to develop solutions, and in this case, one of those is the concept of wetlands, that it also exists in nature. I mean, there is natural wetlands, so we can see it here, but also to take that idea from the nature, but now made it by engineering approach, which are constructed wetlands, a passive technology, I mean a man-made constructed wetland complexes, which takes those nature inspiration, but we can just engineer those systems to treat different types of water, including the industrial or even mine water. We can use it if we can develop the prototype or the design of those constructed wetlands. And what we can see in the wetlands, just as a nature, is a substrate, vegetation, and water. It can also be just like a landscape. This technology really is very good, because we can also made it by engineered tools, look very well using vegetation according to the zone, also to the region. I mean, we can use different types of vegetation and try to construct this technology to treat different types, I think, of water. And one other example, what we are trying to do is using those approaches to implement it in those scenarios. As you can see here, those like lagoon, and we can try to find or to construct these wetlands here to try to avoid to look at this as a pollution, but try to look at the landscape and treat the water to avoid those problems with acid mine. So we are trying to develop, to engineer the system to be implemented on those areas so they can be implemented and try to find good results with those systems, man-made systems. So that's one example of the application of these constructed wetlands in this case. The third case that it's also theoretical that we are on development is the use of one material called biochar, that it can also be used in the constructed wetlands as the substrate, but it can also be used alone as an absorbent material for metal removal, for example. And one of the advantages of this material could be that it can be obtained from different biomass production. So I mean wastes of biomass. It's not to use food now, it's just to use a waste, no, agricultural waste. So it can be obtained from different types of biomass, including municipal solid waste, food process waste, agricultural residues, sludges from wastewater treatment plants, and then use pyrolysis to obtain the carbon-rich material, which is a little bit like carbon activated. It looks like this, but it has another properties, and also but it looks like carbon, activated carbon. So one of the benefits that could lead to biochar use is also the waste management. It can also lead to energy production. It can be used for remediation. It can be used in soil amendment to enhance plant growth and many things more. And also because it also helps for mitigation of carbon emissions, since the main resident time of this material is around hundreds of years, so thousands of years. So it can be used for different approaches in environmental, and this material is taking in the last, I mean, like 20 years, a lot of efforts to trying to use it in different approaches for remediation, but also in different technologies to take advantage of these materials. And some studies are just in development. So we are trying also to use it and to make our effort to implement this material in other technologies, and looking also for the interaction with microorganisms that we are taking. So in the last case, what I'm here just presenting is like a combination of technology that can lead to a good approach in the treatment of industrial effluents. For example, the biological synthesis of nanoparticles, which can also be achieved using a pure strain, or also a consortium, or I mean a group of microorganisms for the treatment of waste streams that we contain some metal. In this case, it was palladium coming from an industry, and using a bioreactor an inoculum of anaerobic microorganisms, I mean, which can be methanogenic, but it can also be a pure stretch. We can lead or promote the reduction of palladium 2 to palladium 0, that it can be recovered as a nanoparticle, which is also just promoted by the microbial activity. And once the palladium it is reduced and form the palladium 0, it is a catalyzer of different reactions. So we can use these nanoparticles for the development of different other reactions, from the halogenation, nitro reduction, hydrogenation, and all of those reactions that can also be used to treat another contaminant for the treatment of other, or for the treatment or in another thing. So this approach is the treatment, but also the recover and revalorization of one metal by using microorganisms that can use here. And this also a good example of how the engineering approach can be used to develop solutions that can be or that can lead to more to other treatments. So as in this case. So yeah, conclude. So what we need to know, of course, is the way that we can develop those solutions. So the know-how of those scenarios is essential. I need to characterize the sites. I need to know the microorganisms activity. I need to promote some reaction, while I need to avoid other reactions. So I need to have a clear mind and to know what is the best part of the route of the pathway that we could lead to the results that we are looking for in environmental applications. So we have a deep understanding of those mechanisms, not also the biochemical, but also the chemical, physicochemical, mineral, and all of those related to this. So this they require a lot of knowledge on different areas also. So I'm talking about the first part, of course, the isolation, the enrichment, and try to find microorganisms able to be used on environmental remediation. It's crucial. We're trying to find in different parts of the world, trying to use microorganisms, trying to isolate it from nature, trying to modification like genetic or biomolecular techniques, and all of those tools that we can use now to try to make biotechnology a better way or a tool to find solutions in different ways. So more of researchers are trying to find those microorganisms, maybe by genetic modification. Another thing is the engineering of the process. I mean, engineers they are a crucial part, because try to find a microorganism, but then to try to find which is the best way in a large scale to be implemented. It is another an important part of the job. So we need to develop reactor design and all of that. It implies the use of the process to try to imagine how the influent would be treated, how the effluent will be characterized, and all what is happening in the middle of the process. The development of those steps, it's made by engineer knowledge. So, and also the monitoring and experimental and research are very related, because we need to develop in the first stages, maybe basic steps like try to lab scale experiments, control conditions, analytical techniques, before try to make large scale or field experiments or long-term experiments. So we need to try to make step by step, but based on those results that we are obtaining from those early phases. And at the end, what we are trying to look to find is or develop remediation strategies, but remediation that can be sustainable, not just in one way, but more than one. I mean, it's not just environment, it is economic, it is social. So we are trying to develop or to look at those factors as well. And of course, in the meantime, if one or more of the metals can be recovered, it will lead also to an added value to the research. We are not just trying to remediate, but in some cases we can also be focused on the recovery of the metals, which could lead to economic value also. So it is some of the conclusions I can show you and share with you. Thank you for being in this speech.
Thank you, Dr. Pat Espadas, for the interesting talk. Now we're going here into the live audience here at the auditorium. If you have any questions, guys, any at all, you know, it was a very, very, very complex topic with a lot of angles attached to it, you know, not only the chemical, but the biological, the economical, the social. Any questions, guys, about this kind of project?
I would like to know in which cases it is useful to immobilize potentially toxic elements, and in which cases it is useful to mobilize them.
Could you hear that, Doctor?
A little bit. I just try about mobilized and immobilized treatment. Which one to choose?
Yes, they're going to repeat the question, but closer to the microphone.
I wanted to know in which cases it is useful to mobilize the potentially toxic elements, and in which cases it is useful to immobilize, or is it only useful to mobilize them?
Okay, no, it's such a good question. It depends on the scenario and on the characteristic of the site and also of the waste. If we can, let me give you an example. I think, for example, some cases, solid wastes can be, think about mobilizing which is in there. I mean, for example, leaching process is about that. We can just to take a part of the solid, the metals, and so we promote immobilization by leaching, which is also called bioleaching, and it can be achieved by microorganisms such as fungi or bacteria. But in here we are trying to transfer from the solid to a liquid phase, and in that case we are using the mobilization approach, which can be also useful for mineral extraction from in mining activities. But there are also another scenarios when I'm mobilizing, such as those that I presented to you, which is it can be wastes which they have no value, or they are too much that we cannot use it, the bioleaching, that we prefer to immobilize them. So we try to promote those reactions. So it depends on the scenario. We need to look at which is the better, and we can achieve in situ or ex situ. I mean, on site or out of the site. If we do need a bioreactor, if we need to find out the cost, and also what are the products that I will obtain. I mean, it's something. It's a metal that it can have an added value, or it is a mix of metals that with or just can be another problem. We need to study that situation to find out which is the better.
Thank you.
Thank you.
Any other questions, guys? No? I have one. Doctor, you showed one of your projects some results before and after the bioremediation process, right? When we use some microorganisms. Could you tell us the time between the before and the after? Are these processes time consuming? Are they relatively fast? What's the time margin we're talking about?
About the time that it took Well, it's such a good question also, because as I have a little time to present it, I avoid, or maybe I forgot to tell you something, that it was that we tried with the without an added inoculum. I mean, just the endogenous biomass that could be found on the samples, and it takes too long, more than a year, to find sulfide reducers, microorganisms on those samples. But we also tried the treatment by adding an active inoculum anaerobes, and that approach, it takes of course, it was a quickly treatment, because it takes like I know, like a week to achieve those results. In scale of time, it's a quite different. Try to find out endogenous microorganism, try to isolate microorganism, or add an active inoculum. But for biotechnological approach, can be used both of them. But if we can use the first one, which takes too long, what we need to do that is to enrich the culture, and maybe after that we will be able to short the time of the treatment.
Okay, that's very interesting. That's very interesting. So another question, and this is, I've been curious about it for some time. Is there any database here in Mexico about affected areas with PTEs, or is not, or there's no registry, and it's very difficult to have this information available to everyone?
Yes, I think that I'm not aware about that database about those measurements in environmental samples. The one that I presented also regarding historic mining waste, it was very hard to construct the map and to try to find out where and what metals are contained on those wastes. There are also just academic studies, and we need to search on the web of knowledge or something and try to figure out and to put all of the information together. We try to do that, but there is also an effort on construct a kind of that database by the government that is still in work, and I hope that in this year or in the next one, those results will be published. Maybe that information will be of a great opportunity to know what is the scenarios in Mexico.
Okay. Yeah, thank you very much. I think that would be a good opportunity for young biotechnologists and to have this effort and mapping all these damaged areas here in Mexico.
There's time I'd like to ask something.
Yes, there's another question.
All right. Can you hear me from here, or do I have to approach the microphone? That's the first.
That's the end.
Hi, can you hear me?
Yes, so clear.
So I'm not a biotechnologist, but I'm curious about the funding of these efforts, because it's a large problem, and I know that public funds are going heavily, or at least I hope heavily, into these type of efforts. But I was wondering if there was private interest in this bioremediation techniques, or to your knowledge, are there any privately funded efforts in this direction?
Okay. Yeah, another is a great question, and okay, from the best of my knowledge, I think that there is not such a. Oh yeah, there is some opportunities, of course, to try to get funding from government or institutions by Mexico, but Mexican institutions. But private funding, there is also not as easy to get as it could be. So what we are trying to do is to develop or to contact those persons. But what we need first is to characterize the sites, to make the situation like be like the focus of the attention. I mean, because there are a lot of places that are contaminated, but people don't know. People doesn't know what is happening with them, the problems. So I think that it needs to be discussed in the next levels to achieve so that to attract that attention, because funding also there is quite difficult. There is no like other countries such as USA when there are a lot of fundings to try to remediate places, because remediation takes a lot of money. I mean, we are trying to do it just at the lab scale, but we know that to implement the remediation strategy at the site, it would cost a lot. So I would try to do more efforts on that way. So I'm not aware about those funding.
Thank you.
Okay. If there are no more questions, then again, please. Doctor, we will be contacting you in these next few days to let you know about the document for participation. So we're going to be sending it to you by email. Okay. As far as I know, that's how we're going to do it, because right now it's very difficult to do it physically.
Yeah, don't mind it. Electronic would be good, and thank you so much for the invitation, and I hope you can enjoy.
Wait a minute. My audio. This one. Okay.
Okay.
Can you hear me now?
Oh yes, now I can hear you. Yes.
Muy bien, doctora, le mencionaba que en estos días la vamos a contactar para enviarle la constancia.
Ah,
¿sí?
Sí, claro, no hay problema. Está correcto.
Y me gustaría también ver si esto es alguna oportunidad para establecer algunas relaciones, si pudiera, siempre andamos buscando la oportunidad para que alumnos de nosotros hagan sus prácticas profesionales, sus servicios en otros lados. Entonces,
Okay.
si llegara, sabemos que usted está así un poquito lejos, pero no importa, eventualmente muchos muchachos aquí de la universidad tienen que hacer sus prácticas profesionales y servicios sociales en otras partes del país, o incluso algunos se van al extranjero. Entonces, si a usted le interesa y pudiera ver esa como cercanía,
Sí.
podríamos verlo a futuro. Eso estaría bastante interesante y padre.
Claro que sí, con todo gusto. Yo con muchísimo gusto los recibiría. Claro que sí.
Perfecto. Entonces, aquí ya estoy.
Sí, perfecto.
Y es que estaba aquí ahorita jugando con la cámara aquí del auditorio, porque la verdad no le sé mucho esto. Entonces, bueno, muchísimas gracias. Déjeme agradecerle de nuevo. Fue muy interesante su plática. Yo creo que fue muy enriquecedor para nuestros alumnos.
Ojalá que sí.
Sí, claro que lo fue. Entonces, como le decía, en los próximos días la contactamos, le enviamos la constancia y esperamos poder tener acá una relación de colaboración.
Claro que sí, lo que necesite ya estoy al pendiente, ya por ahí tienen mis datos. Para mí siempre es un gusto. Ojalá que de verdad les haya servido un poco, que haya podido expresarme bien, y con todo gusto, igual con ustedes, lo que necesite, igual si alguna colaboración futura o a corto o mediano plazo, mire, yo con muchísimo gusto, para mí de veras es un placer. Cuando supe que me contactaron, dije, ¡ay, qué padre! ¿No? Yo estoy muy emocionada. Entonces, para mí el gusto es bastante, créame, disfruto mucho hacer estas cosas y trabajar. Entonces, pues, lo que se ofrezca, ya por ahí estamos en contacto.
Perfecto. Muchísimas gracias, doctora.
Un
Entonces,
gusto.
nos estamos viendo pronto. Gracias.
Claro. Hasta luego.
Hasta luego.